In honor of Women's History Month, we're celebrating the women who are helping drive progress in blood cancer research. Today, we're proud to speak with Dr. Glytsou, the recipient of the Children's Leukemia Research Association's 2026 Research Grant. We look forward to discussing the focus of your research, as well as your journey and experience as a woman in the field of blood cancer research. Thank you for joining us today.
Thank you so much for this opportunity to discuss with you our research, and I've been honored and very grateful for this support by the Children Leukemia Research Association.
Yes, thank you so much. Yeah. So could you just give us like a brief overview about yourself?
Sure, yeah, I can give you some background about myself. So I am originally from Greece, where I studied biology in the National Kapodistrian University of Athens. And I have been fascinated always by biology and how like small molecules come together to form a cell, to form an organ and an organism, and how complicated this process is. So I obtained my biology degree back in Greece, and then I wanted to pursue a PhD. And this is when I moved to Italy, to the University of Padua, where I performed research on mitochondrial biology. And I obtained my PhD in biochemistry and biophysics. Really importantly, that University of Padua, is like the second university in the world, and it was where the first woman graduated from a university. So that was definitely an inspiring place to perform graduate studies. And then, though, since early in my studies, I had a particular interest in cancer research. So my goal was to like merge my expertise in mitochondrial biology to and apply it in cancer research. So then I moved to New York and New York University, the medical school, where I performed my postdoctoral studies. And this is where I worked on blood malignancies and trying to understand the mechanism of resistance to some new therapeutics in leukemias. And after that, I was fortunate enough to manage to obtain my independent position in 2023 in the University of Rutgers, where I joined the Department of Chemical Biology, School of Pharmacy, and the Department of Pediatrics and the Division of Pediatric Hematology Oncology in the Medical School of Rutgers University, as well as the Rutgers Cancer Institute of New Jersey. So it's been 3 years where I have been establishing my independent research program, focusing on mitochondrial biology and their role in the progression of different blood malignancies. And it's been a great time, and definitely I'm very grateful for the support from your organization and others to help establish my laboratory.
Definitely.
Yeah, so actually, yeah, I didn't mention, but already in my undergraduate studies back in Greece, I performed some research as an undergrad student in Democritus Cancer Institute, where actually I already worked in blood malignancies. I was focusing on chronic lymphocytic leukemia and acute myeloid leukemia, where we were studying the chromosomal aberrations in the DNA of patients that present these diseases. So I was already particularly interested in the fact that they are like very common diseases, and they are very complex also. So there has been a lot of heterogeneity. So while different people who have leukemia, still there will be different genetics and different cytogenetics presented in these patients. So that made it definitely very fascinating from a biology point of view, but of course really hard to treat. So that was my first experience with working with actually patient samples that we were obtaining in this institute. We were performing the karyotyping, and I would see actually the chromosomes and how these chromosomes will lead to the aberrations will lead to the disease. And then with the years, I mentioned that I studied mitochondrial biology, and it was like for many years the dogma was that mitochondria do not play key roles in the cancer cells, but instead it's more other ways of producing energy that will be more prevalent in cancer cells. However, during the last years, this dogma changes, or it makes the picture much more complex. So we see that some cancers really rely on mitochondrial biology, on both the energy production from mitochondria, but also for other mitochondrial functions. And one of these examples is acute myeloid leukemia, which I studied during my postdoctoral studies in Italy. And we see, and it is now well established, that this type of leukemia depends a lot on mitochondrial functions. It's something that we are now studying on how exactly these structures within the cell play a role in the progression of leukemia and even in the development of therapy resistance.
All right. So what does your typical day in the research lab look like?
There is not a typical day. Every day is a different day, but that makes our profession so exciting. One thing is that, we have so many different tasks that start, and the most important is training and mentoring students and trainees in the lab, but also being part of a faculty community in the university. Being part of different meetings and different committees. And of course, that includes also a lot of reading of the new findings in our field, and a lot of grant writing, which is applying for different sources for support for our research. So depending on the different deadlines and the different schedules, that also I forgot to mention, teaching is a part of my job as well. Teaching undergraduate students in the School of Pharmacy, as well as graduate students at Rutgers University. So a typical day can include any or all of those tasks, and So it will be a combination of a lot of meetings, writing, reading, interactions with colleagues. Actually, to tell you the truth, I'm missing a little bit bench work. In the first years of my independence, I would still be on the bench and perform experiments and teach my trainees how to do that. But now there is much less time left for me being on the bench, and this is now what my students and trainees do mostly. So acute myeloid leukemia is one of the most devastating blood malignancies nowadays. It affects both adults and children. It is more prevalent in adults, but also children can present this malignancy. Although there have been so many advances in understanding this disease pathogenesis, still the treatment options are not doing great. That is shown by the fact that the overall survival is pretty poor for patients with AML. And most importantly, a good percentage of patients, both pediatric and adults, would not respond to the frontline therapies, and or they will develop resistance to the treatment, even within a few months after receiving the first treatment. So that makes it really, really hard to treat disease, and especially for the cases of high risk AML, and also when we have a relapse of AML, this is much more difficult to treat, and there is definitely an urgency to find new therapeutic options. So there have been, as I mentioned, a lot of advances in understanding the disease, and that has led to many new therapeutic options for patients with AML. Many of these new drugs and medications are targeted therapies. That means that they specifically target something that is um, particularly high or specific in the malignant cells, in the cancer cells, and it's not so important in the normal cells. These targeted therapies definitely have given a lot of promise because, unlike chemotherapies, they're more specific treatments and they would cause less side effects. But again, the issue is that unfortunately the patients may develop resistance to these targeted therapies. One of these very promising new targeted therapies is called venetoclax, which is one very encouraging oral medication that specifically kills the malignant, the cancer cells, the leukemia cells in AML. And now this medication has been a breakthrough and is really becoming the standard of care for patients with AML, specifically for adult patients. And given this promise, now there have been many clinical studies where venetoclax is being tested also in pediatric patients with childhood leukemia. And this gives a great initial responses. It seems to be a very tolerable medication and helps eliminate the disease. However, again, the challenge is that we see that patients will still have their leukemia coming back after some time. And the main focus of our research is to understand why leukemia comes back, and even to find some ways to understand why some patients will respond to this targeted therapy or not. And again, because of my special interest in mitochondrial biology and because of our recent findings, we focus on how mitochondria or these subcellular structures within the cell affect and help the acquisition of therapy resistance to these targeted therapies. We in this proposal that has been supported by CLRA, we do very fundamental biology research to find again why mitochondria contribute to the development of therapy resistance. What are these key proteins on mitochondria that drive resistance, and whether these organelles are adequate to drive therapy resistance. Just to mention that a lot of previous studies have focused on like changes in the DNA, so different DNA mutations that may affect the therapy responses. And of course, these are fantastic studies because they have let us understand which genetic populations may respond better to some treatments versus some others that will not respond. However, the novel aspect of our studies is that it's not only the DNA changes, it's also those organelle structures that change, and they actually change their shape in order to support this development of therapy resistance. So we look at we focus on some different subcellular structures in this study that we think that they play key roles biologically in the acquisition of therapy resistance.
Mm.
Yeah, definitely. I was very honored to receive this award by the V Foundation, and definitely was very uplifting to see that the women scientists are being supported uh, by these foundations, and I'm definitely very grateful to the donors and to the associations who have been supporting our work. It was also in the beginning when I was establishing my independent research program. So it was a great support to help um, establish everything, all the experiments we're doing in the lab, but also recruit trainees in my laboratory to do this very fundamental research. Uh, so it has been definitely uplifting, and I have been very honored. And I think it is inspiring, and it's also it can inspire the new generation of women scientists. I do have definitely also young women scientists in my laboratory, whom I hope that they get inspired, and I train them and mentor them in order to help them be successful and do new discoveries in our laboratory and overall during their career. So it's been fantastic to support women in science, and definitely a diverse um, population of trainees, because everyone is needed in order to have success in our discoveries, and these discoveries to eventually move into translation and to our patients.
Mhm.
Yeah, that's wonderful.
Yeah. So as a woman in STEM, like what kind of advice would you offer, other young women who are interested in going into research?
Like any path, nothing is very straightforward. So for each person, the path may be different. And I had my own path, but for another person, it might be different. So definitely, it needs a lot of resilience and patience to be into STEM and be able to ask bold questions that nobody has asked or addressed before, and work in research environments that require a lot of hard work, a lot of, as mentioned, resilience and patience, and it's it is not an easy, straightforward path, but you need to be patient to fulfill your dreams. Definitely asking for help around from colleagues, both women and men, obviously, and finding the key mentors is very, very important for a career trajectory in research. Uh, as I do now with my mentees, I try to spend a lot of time tailoring their mentoring approach and the training plan. This is how this happened with me while I was a mentee with my previous mentors. But not only the mentors in our laboratory, but also I would advise to seek for advice and mentorship from other colleagues or other people around that can be in the university or out of the university. And I, another, yeah, maybe some other advice would be to plan and think in advance so you can achieve your goal that you want to achieve, and yeah, be surrounded by a great support environment and great mentors. Very important.
Yeah,
Yeah.
absolutely. I think it's like, yeah, very important that you have a support system that, helps you encourage to go into a field, a more male-dominated field, so it's very cool. Yeah.
Um, and so going to the next question, what are the main differences between pediatric versus adult acute myeloid leukemia care and treatment? Does a patient's age influence treatment decisions? I know you touched a little bit on that earlier.
Yeah.
Right. Yeah, there are differences. There are genetic differences that are found between children and adult acute myeloid leukemia patients, though some of the same genetic mutations may be found in both groups, but they may be more prevalent in one group versus the other. Um, and however, yeah, still acute myeloid leukemia is a very particular disease because it is very heterogeneous. So even within the childhood population or within the adult populations, there are so many different subtypes and cases. So what it's really, eventually, it's really like a disease of a person, and eventually we want to achieve something like more of a personalized medicine. But again, yeah, as I've mentioned before, indeed the therapeutic options are different now still between adults and children. And some of the targeted therapies that have been now started being used in adult patients, they are not yet used in the pediatric population, but they have been tested. Um, Again, the common issue is when we have relapse. So even a relapse after chemotherapy or the standard care of use, or relapse upon targeted therapy, in both the age groups, that makes the situation much more dismal. And this is where we focus our research on, both in pediatric and adult AML. Right, there is a combination of chemotherapeutics that are used.
Mhm.
But again, as I mentioned, even in the adult population, the children populations, there is a risk stratification of the patients
Yeah.
that then would design the specific treatment that the AML patients will receive. And then it has to do also with the genetics of the patient. So nowadays we can indeed check if the patient have specific mutations, and we have specific therapeutics for those mutations. For example, the FLT3-ITD mutation or an IDH mutations. We have some specific new targeted therapies that show promise for these specific subpopulations. And then of course it is another of course other risk factors like the age, prior diagnosis, whether this was the first presentation of leukemia or whether it was a different myelodysplastic syndrome or other pathology that was essentially developed into AML. And lastly, there is the option of transplantation for some patients that are fit. That, of course, that's a great option, but still there is a need to find matched donors. And also there, this may also have some side effects. So yeah, definitely we have a lot of options to choose from. But again, there might be still some challenges with all of those different treatments. It is a very difficult situation that again the patient needs to have a great support system that will, of course, derive from the clinicians, the nurses, psychologists within the team of the clinicians. And I think it's really, really important for the patient to understand that each AML is different. It is very heterogeneous disease, and each patient, based on the history, they are different genetic mutations. They may have different options for treatment, and it is definitely very promising nowadays that what research has managed to achieve to have so different treatment options and more specific targeted therapies that would eliminate the side effects. So it is definitely very encouraging to know that. But yeah, it is something that people have to understand, that it's a more personalized treatment approach, and it might be different from other patients with AML.
So, how does Children's Leukemia Research Association's research grant support your work, and what outcomes do you hope your research will ultimately achieve?
Again, I'm very grateful for the support because particularly our proposal and our project is really basic research, and we want to address basic biology questions that will provide this fundamental knowledge to better understand the mechanism of therapy resistance. So why some people respond, why some patients will not respond, and then also find new targeted therapies or new targets within the cells that will help circumvent or tackle this therapy resistance. So with our proposed project, we ask some new questions on how mitochondria play a role in this acquisition of therapy resistance. And we want to, upon successful completion of this project, we hope that we will have a much better understanding how therapy resistance works. We might have some new biomarkers or some new indications on when a person will respond or not respond to some specific targeted therapies. And lastly, again, we will propose some new targets that we can later on work with chemists that we can specifically target with some new medications, new chemical compounds, and propose maybe some new combinatorial treatment for the difficult to treat cases of AML.
Yeah,
It's very
that's amazing.
exciting.
Yeah.
Very cool.
And wrapping up our questions, you may have just answered a little bit about this right now, but looking ahead, what gives you the most hope about the future of AML research, and treatment options for patients moving forward?
Definitely, we live in exciting moments. And now we have amazing technologies, really uh, advanced technologies, for example, new microscopy approaches, new technologies to study leukemia at the single cell level, so in every single cell of the leukemia of the patient, and even other approaches that we use in the laboratory, like the CRISPR screening approaches, and many more that we and others use in our laboratories in order to really dissect and understand how the leukemia evolves. And I mean, that's very helpful in all the meetings that I'm attending nowadays. I know that there is a lot of new knowledge that was not like 10 years ago. Again, new technology supporting these experiments. And this really accelerates the drug discovery process. And again, it is clear to all the scientists that we need more tailored therapeutic approaches for more personalized medicine. So we are going towards that direction, and that is really helpful for the patients with acute myeloid leukemia. Again, in the last 5-6 years, there have been many different new medications that have been approved by the FDA because we understand much better leukemia, and I believe there will be many more, and we will know which patients will benefit the most from these different tools that we have nowadays. So it's been definitely a very hopeful period, and we hope with our research that is supported by CLRA, we will add our own knowledge into this big puzzle of the complex cancer evolution.
Wonderful. Thank you so much. And do you have any last words you'd like to say before we conclude our interview today?
Yeah, again, thank you so much for this great opportunity to discuss with you and with your audience and the donors, the research we are doing. It is really fantastic to get support for this very basic biology that we perform in the laboratory, and especially supporting women in STEM and young investigators like myself. Uh, Very grateful to the donors and the association. And it's fantastic work, and it's really, really needed nowadays. Thank you so much.
Thank you,
Thank you.
Yeah, thank you so much, Dr. Glytsou, for taking time to speak with us today, and for the meaningful work that you're doing in blood cancer research. We look forward to following the progress of your research and the impact it will have in the years ahead. Thank you.
Thank you so much.
Thank you again so much.
All
Thank
right.
you.